Hi everyone, and welcome back to the Bio Student Project. Today we will be talking about transcription and translation. Transcription and translation are key processes in the synthesis of a protein. First comes transcription. Transcription is when an enzyme called RNA polymerase uses DNA as a template strand to make messenger RNA, or mRNA. There are 3 stages of transcription: initiation, elongation, and termination. In initiation, RNA polymerase attaches to the promoter sequence of the DNA and separates the double-stranded DNA into 2 single strands. Next is elongation. Recall from the previous videos about base pairings. A goes with T and G goes with C. Well, in RNA, there is no thymine or T. Instead, RNA has uracil, abbreviated with a U. So in RNA, C goes with G and A goes with U. Then in the last step, termination, the terminator sequences in the DNA signal that the process is complete. From there, the mRNA is completed and finalized in bacteria, but in eukaryotes, it undergoes an editing process. The ends of the mRNA are modified and a 5' cap is added to the beginning, while a 3' poly-A tail is added to the end. Additionally, parts of the mRNA are cut out of the sequence and the broken up chain is glued back together. The sequences that were removed from the mRNA are known as introns, while the pieces that were glued back together are known as exons. Now we are ready to move on to translation. Translation is when a polypeptide chain is built by a sequence of amino acids. Groups of 3 nucleotide bases, known as codons, code for specific amino acids. First, tRNA, or transfer RNA, takes the amino acid, methionine, and it attaches to the start codon, which is most often AUG. There are more details about this step, but for simplicity I will skip it in this video. Anyways, more amino acids are coded for by the codons, and you eventually have a polypeptide chain of thousands of amino acids. Finally, the process is finished when mRNA reaches the stop codon, which is either UAA, UAG, or UGA.